Today we have one of the winners of the OpenVT 2025 shareable model competition. We're very glad that Emanuele Scacchi is willing to talk to us today about that. We'll have a little bit of housekeeping before we get to the seminar proper. As always, Tom Helikar and myself, Jim Sluka and Lorenzo are available to take your suggestions, comments, critiques. Please let us know what we can do to make this a more useful series to you. We have a variety of communication channels. They work better if we use them. There's a Slack channel, the GLIMPRINT website, the IMAG MSM wiki page, a LinkedIn feed, the YouTube channel, and so on. Today, we're going to have a seminar on small mobile RNAs and Turing patterns. We'll get to that in a second. If you have a publication to share, please let us know. We could put it up on the IMAG page and also on the GLIMPRINT homepage. On the GLIMPRINT homepage, you can also list meeting announcements, seminar announcements, workshops, classes, job openings, funding opportunities, and so on. If you have any issues with that, please contact Jim Sluka. We'll do our best to get it up for you. Coming up November 6th, on a non-standard date, there is a talk on mathematical modeling of beta cell turnover. We have a talk on November 13th at a non-standard time by Shihao Zhang from Georgia Tech. November 20th from Columbia. November 27th is a holiday here in the United States, so there's no meeting. And then we have Emilia Luca and Job Berkhout continuing during the season. If you have people you'd like to hear from, you want to suggest yourself, your students, your postdocs, or people who've written papers, experimental, computational, or otherwise, that you'd like to hear, we'll do our best to get them on the schedule. Please reach out to us for that. We will not have a steering committee meeting tomorrow. So today, again, we have one of the winners of the OpenVT shareable modeling competition from 2025. Emanuele Scacchi will be talking to us about mobile small RNAs and Turing patterns, coordinating leaf polarity and shape diversity.
My name is Emanuele Scacchi. CH, like Machiavelli, is changed sound in Italian. But yes, a part of this, I am delighted of this invitation. I want to thank you, everybody. And thank you, all the people that joined for following me in this seminar. And yes, the title suggests that I'm going to talk about the small-RNA Turing pattern. But I would like to break the ice with this painting from Picasso. And in the end, what I want to point, it's something apparently far from biology. This should be a vase of flowers on a table, and we see how Picasso go far away from the reality of what we see. But essentially what I want to point is that when human creates, we first imagine, after we design, and after we build, and we have essentially this algorithmically mind, an engineering approach to build and to create. And today, the talk is on a completely opposite side, is on self-organization, and, in contrast to the Picasso painting that will produce astonishing order, shape, forms, without any architect, without any design or creativity, shape, color, symmetry, just emerge spontaneously. And these emergences come from interaction at local scale that could be between molecules, between cells, between individuals, or between population, and the all over order essentially spontaneously rise. Today, I want to talk about one of these self-organizing examples, because the difference in leaf shape is pretty remarkable. And despite their diversity, here you see, can you see, I hope that you can see my arrow, and you see the flat leaf or Arabidopsis thaliana. But such flatness could change between relative species or between the leaves of the same species, from flat to radial to bifacial, or to produce a complex shape, like in the case of carnivorous plant. So yes, essentially, in this talk, I want to use the leaf as model system to explore in deep the question of how can genetic network self-organize a pattern in nature, and especially here I am interested on the pattern underlying the diversity of the leaves that you see here. You essentially plant to make a flat leaf has to perfectly position in two domains that are called adaxial, abaxial. And the difference in the shape between these leaves is the different positioning of the genes underlying this process. So essentially, I want to look for the organizing principle of this system, and we can suppose that each cell contains the same genetic material and the same program, and this program autonomously, it should be able to self-organize, in this case, polarity, symmetry, or the pattern underlying the diversity of the leaves. So first of all, how a leaf originates. And plant, they can continuously grow through their entire lifespan. Some plants can grow for 100 years. And this is due by two populations of stem cells positioned on the opposite of the plant. On the bottom, we have the root meristem, and on top, the shoot apical meristem. Such population of cells essentially contain stem cells, and they continuously produce daughter cells that can allow the growth. Here we have the central zone of the meristem that control the activity of the peripheral cell that proliferate, and eventually a lateral organ, in this case the leaf, can rise. And from the meristem to the tip of the leaf, you can see on the right, we can distinguish symmetry just from proximal to the meristem to distal, from the lateral part we call mediolateral, or the top and the bottom, that as I anticipate we call the adaxial or abaxial domain. And what's the difference between adaxial and abaxial? Essentially, the formation of this domain characterizes the cell type and the polarity of the organ. Without entering too much in detail, what is really interesting, and this is on the right, I place an image from the paper of the lab of Enrico Cohen. Is a fantastic paper where they use essentially a top-bottom approach. They asked themselves which could be the constraint, that can make from a dome shape that we can imagine that is the primordium of the leaf. We can generate a flat leaves, or we can have a needle shape, or more complex structure like the trap of carnivorous plant. Essentially, what we need that two domains should be counteropposed, and this distribution of difference in constraining the force can make possible to generate the flat leaves. So the question here that I'm going to pose to myself is on the other way around. I wonder how it's possible to generate such domain in the leaf. Now, we go more experimentally. I show you the shoot apical meristem of Arabidopsis thaliana. You see here what you can see here is a time lapse, following over the time the growth of the leaf, the morphogenesis of the leaves. We see that the cells are specified before we see the dome, the shape emerge. This is a 72 hours time lapse. And essentially, the genes that are responsible for the polarity of the leaf are already, they form polarity already in the meristem. The leaf is specified on top of a polarity. And later on, the leaf from such pre-established polarity somehow acquire independency and it starts to grow. And it is here I show you a small network of genes where how it's possible to coordinate these two domains that establish the top and the bottom of the leaf. Essentially, there are small RNA, a microRNA, and a thaliana that are implant are able to diffuse between cells. And so when a molecule diffuse is produced from a cell, it can travel around. Essentially, it can coordinate the behavior of the surrounding cells and between cells. So starting from these, the idea was, as anticipated, the idea was, can we understand, can we establish the organizing principle of this process? And we have in mind essentially two main mechanisms that has been already discovered in biology. You know, just by zooming out, we have the Drosophila embryo, where essentially the Drosophila embryo maintain the same size, and there is a strict hierarchical cascade where that produce essentially the stripy pattern of the embryo. And this is a pretty rigid system. On the other side, we have a more dynamic system that this is the example of the left-right pattern in zebrafish embryo, where nodal and lefty essentially already know somehow the polarity by the context in which the embryo is placed, and they can reinforce this polarity while they grow by essentially self-organized on so-called reaction diffusion mechanism, essentially what we call now what is well known as a Turing mechanism. And for the leaf, essentially we immediately point that rigid system like the case of Drosophila, more grounded in what we can call a positional information, that's not really compatible, because the leaf, essentially, as you see from the meristem of Arabidopsis, essentially start from a fixed polarization, but after it move, because the meristem grow, and it start to grow the leaf, so the leaf start to be far away. The cell change position eventually, or changing the organ changing size. So we thought that more dynamic system could be compatible also because we have this small RNA that can diffuse, and they essentially react with transcription factor, and we have transcription factor that react between each other. So we have a high chance that the system could be essentially a self-organizing system through reaction diffusion. So what was the approach? Essentially, we set up something like a high throughput theoretical biology approach. We have these guys that you see on the left. That's not really important to think about the name of this guy, but these are this node that I draw here. Essentially, are family of genes. We have HD-zip genes, AS2, AS2-like, CAN, ARF, and we have two molecules, two micro, that two small RNA that can diffuse between the cells. So essentially, the first step was to formalize the system through essentially how we do that is a system of partial differential equation with two diffusible molecule, and after we apply the mathematical principle of the Turing morphogenesis, because when we talk about Turing pattern, when we talk about this Turing pattern that are made through a Turing-like system through a reaction diffusion, what Turing did, essentially, it set up a theory. Theories make out of theorems, and then you end up with a series of constraints, and the constraint that could, in the case of a system of partial differential equation, reflect on the parameters. And here I use what we can call a symbolic reasoning engine, Mathematica, language essentially. And essentially what is possible here? It's possible to reason over algebraic symbols. And in practice, from the theoretical framework of Turing, I ask what kind of topology I can have with two diffusible molecule. And essentially, we end up with 130 networks, but 130 networks that essentially split in space the diffusion molecule. So this is the first rule that we ask. And we have also another two rules that micro can interact only with one target, essentially. So the only among the connection that you see already there, only the red one, the inhibition of the micro of the target, and the split in space of the micro is the only rule that has been applied here. And in the end, we end up with just one network. That here you see that is a three-component network, is a minimal topology. This means that this topology contain the minimal number of connections that are indispensable to and sufficient to create a Turing pattern. And here you see that essentially the genes are grouped in modules, and this network is mainly dominated by negative interaction, apart from the induction. The asymmetric leaves Stasi have a node by the HDZ node. So the first thing until now as was purely theoretical, you know. So what is the first property of a self-organizing system? Essentially, is the test to be able to produce order from chaotic initial condition. So from random initial condition, we run a simple one-dimensional simulation, and is able to create what characterizes this kind of pattern is a heterogeneous pattern and stable over the time. And such pattern has a feature that I will recall during this talk is the wavelength, you know, essentially is the size of the pattern. So the pattern was also tested in a cellular system. This is a static hexagonal stripe of cell that should phenocopy the mediolateral section of the leaf. Essentially, you know, if you cut transversally a leaf, you should see about 6-7 cells as sections. So this should phenocopy this. And from random initial condition, it produces something like we call a variegate pattern, you know. Also, we think about the first paper of pattern of Turing, he showed this variegate pattern that should phenocopy the color of the cow skin. And let's just recall what I told you from the beginning, that the leaf rise on an already prepatterned population of cells. So what's happening if we just specify that one of the guy as prepattern is polar, you know, just the first stripe of the cell, like you see in the blue here, it's enough to make possible that this system self-organize in what we can call bipolar stripes. And as final test, our question was, can this network, a Turing system, a Turing reaction system, self-organize the polarity while the leaf grows, essentially, no? And the answer is yes. And if you run this system, each cell run a following the system in a growing organ, while the growth move on, and the cell may change position, or there could be some change in the context, the system is able to maintain the polarity by reorganizing the pattern over the time. So what we call the leaf polarity model finally is able to self-organize into bipolar stripes. And as test, we also test the same model in 3D if it's able to propagate this bipolarity in a tricellular model, and the answer is yes. So on one side, this is purely a theoretical and modeling approach. On the other side, we wanted to go deeper inside experimentally. So what we did, these guys are transcription factor. Essentially, they bind the DNA and they activate or repress a gene. So the test was to make a high throughput. So for each of these guys, we make a cheap experiment on the promoter of the other guys. What does it mean? We wanted to know if each of these guys physically can bind any of the others. And we discovered that everybody bind the promoter of everybody else. And also we make an induction system for which it was possible to make a pulse of induction of each of these guys and follow the behavior of the other genes over the time. And essentially they almost all connected. And so what we have been able to see, we have been able to see that everybody are physically connected because they bind the promoter of the other and they regulate the other, and we know which kind of regulation by making short-term activation. And we end up with this experimental gene regulatory network that, like the LPM, is dominated by negative relation, but the what we can call red node, the HD zip, is the only exception that induce asymmetric leaves and thaliana. Like in the LPM, HD zip induce asymmetric leaves. Now, if we take the experimental network, finally, to arrive to the leaf polarity model, the theoretical one, we did a system of simplification where we merge nodes in modules. We use the same system for simplified experimental network, and we can see that essentially it is exactly like the LPM. So this theoretical approach was predictive on the experimental data. The only imposed relation through which we start the theoretical approach was the red connection, the micro-RNA repression of the target. For the rest, in blue, you can see that we predict the entire topology of the experimental network. But on top, with the experimentation, we discovered two connections that were not included in the minimal topology. Because just to remember, the leaf polarity model is a minimal topology that can't be smaller, but in the end can contain more connections. So if we simulate these two extra connections, the result essentially do not change. They have only a quantitative effect on the outcome of the model. So now we have another problem, you know. The RPM is a three-component, it's made out of module. What we want to know is essentially which is the minimal experimental topology that can achieve the self-organization of leaf polarity. And we start to deconvolute each node of the minimal topology. And the first discovery was that when you deconvolute the green node, if asymmetric leaves 2, AS2, this guy, do not diffuse, the system doesn't work. And this is a transcription factor, usually are supposed to do not diffuse in plant. Diffusion is not something that it's common for all the protein, let's say, in plant. So what we did, we essentially take a look about the activity of the gene, of the promoter with this first construct. And essentially, asymmetric loop is expressed only in the first layer of the upper epidermis, the top epidermis. But if we check the protein in plant, the protein is present also in the lower layer of the leaf. This suggests diffusion movement. So what we did, if we lock the diffusion of the protein with that big epitope, if we put 3 Venus in this case, the fluorophore that make possible the detection, the protein can't move anymore. And if it can't move, is present only on the top epidermal layer. So this suggests that AS2 diffuse, but is it functionally relevant, this? So what we did, here you see the leaf of the wild type. And if you knock out, if you remove asymmetric leaves, the leaf look like severely affected. But if you transform with the construct, you put inside the protein that we see that it diffuse, it's possible to revert to wild type. But this is not possible if you introduce AS2 that do not diffuse, the third one. So what we discovered is essentially this was a prediction that the model produced the bipolar, if we model, it produced the bipolar stripe only if AS2 diffuse. We show the movement, and the movement is functionally relevant. Also here in the right, you see what we call protoplast assays to test if the protein is functional. There is difference in functionality between two constructs, and there is no difference in functionality in between moving or not moving AS2. The difference is just that it's not able to rescue the mutant. So, and another point was that the only guys that they didn't show any interaction with the microRNA on the bottom, what is B160, was CAN and ARF, you know? And we saw that the topology was essentially unconnected. We started to play simulation about the fact that the organ change inside, and we need a regulator essentially that will be dynamic, and ARF and CAN are kind of, especially ARF, is dynamic in the system. So we suppose that also because we have seen from other papers from other group that CAN and ARF can form a dimer, we thought that could dimerize, maybe they could induce MIR, and so while the leaf grow in size, they can fine-tune the wavelength, the size of the pattern. So what's going on here? Essentially that this is the promoter of MIR, and AS2 and CAN, they bind the same domain physically. And while if we induce ARF and CAN separately, they do not have any effect on MIR. If we induce together, they are able to induce MIR. So we suppose that this guy form the dimerization of this guy is relevant, and the action of each of them has no impact if they do not act together. And to show this genetically, essentially, here you can see the wild type. This is the dome of the meristem, and here you see the beginning of emergence of the leaf, is what we call P0. And here you can also see the activity of the microRNA gene that is on the bottom of the primordium. On the right, you can see what we call P5, a later stage, where the primordium is evident and the microRNA is on the future bottom of the leaf, the future abaxial. It's interesting to notice here that on the left of the P5, there is also the expression of MIR because there is the emergence of one future primordium. If we break, we knock out CAN, or we knock down ARF through an artificial micro, or if we knock down and out both of them, essentially what's going to happen is that MIR disappear in the primordium in later stage, not at the beginning. So, and what is interesting also in later stage, that MIR, it remain expressed at P0, but disappear only at P5. So what do we discover? That this system, it's something like organ autonomous. It what in a classical paper, it was called from the 50s self-determination, because essentially if you take a primordium of the leaves and you put in vitro, the leaf is able to grow and to finish its morphogenesis. So essentially this CAN-ARF module dimer is something like an organ autonomous system to control the wavelength of the pattern. And this just to show in numerical simulation that if you have this guy that act together, they can increase the level, the internal level of MIR in the pattern, but they also can modulate the wavelength. In the case of induction, you can increase the size of the pattern. So this brings us to another question. Maybe many of you remember this famous paper from Murray that after is in color on Scientific America, where there was this argument between the size of the pattern, the wavelength of the pattern, and the size of the system in which it operates. Essentially the size of the organ, the size of the body, compared to the size of the pattern. Yeah, we can see two guppy with different wavelength or nutshell, or more or less we can see the speculation of Murray was that, we have the same operating system that produces a certain wavelength. If in the leopard, I can have different size of the tail, essentially the pattern operates in a different size system, and you can have a different pattern, essentially. So what's going on in the leaf? If we try to make essentially morphospace. You see here in this plot, this black delimited area is the Turing space. The combination of two parameters for which we have a Turing pattern, outside of which we don't have a Turing pattern. We ran 3 simulations on the growing cellular model. And this is the interaction between essentially the Mirka nerve and the HD-ZIC node. And we wanted to see which kind of phenotype we can have. Essentially, we have kind of a robust and bluish wild-type phenotype. Once the wavelength becomes too short, we can have start to have a kind of a shift or a split of the pattern. We start to have a bifacial pattern down to a labyrinthine and completely chaotic pattern that do not resemble any symmetry. If we increase the wavelength, at a certain point, maybe due by the initial polarization, the patterning start to make what we call polar shift up. Essentially, it start to create a spot of the adaxial, the top domain in a central restricted area. And if we push too much the wavelength, the pattern is essentially radial. What does it reflect this? Essentially, if you take a look at the mutant, we have one set of phenotype in the knockdown mutant. We have a wild type with a bottom abaxial or a top adaxial marker. If we start to knock down CAN1, CAN2, for example, we start to see the phenotype of a shorter wavelength because we decrease the power of this interaction, and we start to see split, let's say, top and bottom domains start to be confused, or a shift, like the bifacial pattern that I show you here. But what is interesting, when the leaf fail to properly grow flat, and is the size of the pattern that decrease, we have an opposite effect, because like Murray was arguing, the same wavelength is you make a different pattern if you change the size of the system. So a pattern that it's shown for a shorter wavelength for a normal size organ is coherent with the modeling. But if we decrease the size of the pattern, of the size of the organ, we can rescue or have even an opposite effect. And now I mixed a little bit the paper of from Cohen Lab, another picture from Botany paper, because essentially, we have a unique gene regulatory network that self-organize this polarity in wild type with the polar or bipolar stripes, and they can make flat leaves. But if I increase the wavelength, I can make a trap of the carnivorous plant, or I can make a radial leaf. Or if I make shorter wavelength, I can make a bifacial leaf. So this means that it also explains how it's possible that the same plant can make flat leaves or a carnivorous trap, or the same plant can make a flat leaf or a needle-like shaped leaves, or close relative plants can have different shape. And he has a little bit of a recapitulate. We can have a flat leaf by bipolar pattern. We could have a polar shift up, and we can have a trap, or we can have radial symmetry that could reflect, for example, a needle shape or a radial structure of the leaf. So this last part is a little bit speculative because, of course, we didn't demonstrate mechanically that this pattern underline by modeling the change in shape, but essentially the principle of patterning is there, and this could explain the difference between within and between the species. So essentially we propose this highly interconnected G regulatory network that governs adaxial, abaxial, polarity. But what is cool, we show that small RNA can contribute to the generation of a Turing system to create a self-sustaining or self-organized self-sustaining system that govern in this case, the plurality of thread leaves while a system work. And that could be a very important if you think about synthetic biology, because we are still not able to make de novo protein to create a de novo circuit, but a microRNA is fully programmable. So we can essentially think to create novel micro to create de novo Turing system in plants. And we have also this vision, positional information. I talked at the beginning about Drosophila or zebrafish embryo, but we have a very eminent example where, let's say, a positional information, a fixed information can interact at the same time with a self-organizing Turing system. In this case, we show that we could have a prepattern, and this prepattern can guide the first step of a self-organizing system to, after letting independently regulate the patterning during growth of the leaf in this case. And also, we have shown that such flexible system is able to give rise to several patterning that explain how you could have variation in leaf shape within a species or between species. And I think that that's all for today. And I have to acknowledge the group because I am a postdoc in the Meyer Tinderman, and this work has been possible thanks to the fundamental work of Gael and Poix at experimental level, and also for the help of Shea, Agatha, and also I would like to thank Walter at the back that gave me assistant for this. And I see in the public that there is a loose brush, and I should acknowledge also him because we continuously work, and when you will see the next chapter in the future, there will be also Lutz that will be part of the game. So thank you very much for listening.
Okay, time for some questions. If people need to unmute, put a request in the chat. Some people already have privileges. So please go ahead.
Well, I have one or two questions. So, well, first of all, thank you very much for your talk. It was very, very interesting and nice. I've been working with some similar kind of networks. It's more related to notch, related to cell-cell contact. And there are two things that comes out of your work that I'm curious about. So you show in your network, eventually, if I understand it well, you eventually reach a stable patterning, and so your network stabilized to some results, generate stripes or different domains, right? And I wonder if you have explored areas where instead the system don't stabilize, or if your system can generate some phase system where it doesn't stabilize and keep oscillating. And I wonder if you look at that. And the second one is that in my work, I notice that when it's about things happening between cells that are just opposed, or cell-cell contact, or well, you show paracrine signaling, it might be that the geometry of the cell affects the final phenotype, reinforcing one or the other mode of the network. And I wonder if you observe any of these in your system, if you look at that. And that's it.
Yeah. Very good questions. So for the first one, of course, from a simulation point of view, if you go out of the Turing space, there is a sub-Turing space where you have these excitable media or waves, traveling waves. And they are pretty hard to detect in plant because, and this is also answer a little bit to your second question, because the chemical reaction are much faster than the grow of the plant, and essentially we didn't go really far away because the organs start to be too big, and we know at the beginning there is a prepattern that maybe may reinforce. But I also want to consider that what I propose is just a minimal network, and if you check the supplementary data of the parameter of the model are made for maximize the parameter with value 1, because we don't have really an idea. We say, okay, they are all transcription factor, maybe they all behave with the same kinetic. But the plant, for example, that make a flat leaf, always make a flat leaf. So this means that there are contour things that make the system super robust. So I think that while I could give you some indication that I can make some kind of pattern, maybe the possibility of a traveling wave is not possible in plant because it could affect the robustness of the entire plant. And so in the mutant, because this guy, for example, HD-zip that is and MIR, they regulate several aspects of the development, the root, the leaf, the sexual organs. So I would say that something goes wrong, but the plant should cope this with extremely high robustness. So yes, it's possible, but from a modeling point of view, but from a biological point of view, we didn't see any of these kind of things. For the second answer, I think that you talked to me about Notch, and Notch work in a way of organizing an organizing center, in the sense that one cell, at a certain point, to say I am an organizing center, I regulate the surrounding. So the geometry has something of a role in building, tell me if I'm wrong. In our case, we start from with several shape and from a continuous model, and we didn't see any difference. Also, we have the leaf, few cell really move and could do something in plant in terms of changing position. We have a cellular pot model that is not a very good, is a compromise to have to make possible to have a lot of simulation. It was much faster for us for simulation. But we also set up a system from which we can see when the cell move, when the cell could change a shape that is no more realistic a plant cell. And so we have thousands of simulation. Whatever happened, the cell it's crappy, the cell is hexagonal, triangular, completely. The pattern is always the same, you know. We think that for this simple pattern that is just top and bottom, there is an extremely high robustness in realizing that, and we notice that just the wavelength is the fundamental rule to make it. But what happened to the cell do not matter too much, apart a difference in the interface between on and off between the two patterns.
Thank you very much. Yes, about not
Thank you.
that in neural development, yes, there are disorganizing center. We were asking the other question, in other system like the endothelium, we have cells that are all homogeneous, and then we asked what happens if we put them in a way that they can contact, they have notch, what will happen, what is the pattern that emerges there, and so on. And so it's slightly different, but yeah, I think I get your thing on the plan. Thank you very much.
Thank you for asking.
Okay, Lutz, you had your question.
Hi, Emanuele. Thank
Hi,
you
Lutz.
for actually opening up our eyes, or at least my eyes, for this beauty in the plant world, because we rarely see modeling applied to these developmental questions in the plant world. And you nicely showed how these processes are linked to the discrete nature of the cells, and you have in the model apparently superimposed the reaction kinetics with the cell division processes, and your domain basically grows and cells divide, right? And mathematically, that means there is an additional process beyond reaction diffusion, because when the cells divide, push away their neighbors, which take with them their local concentration values. So actually you have an advection process that is driven by the rate of cell division. And that may give additional effects that you don't expect from the classical Turing analysis. And that's why
a very good point.
a question would be, isn't the cell cycle duration and therefore one of the parameters in the cell-based discretized model also an important control parameter? And one could predict maybe thresholds and say, if the cells divide too quickly, that may also change this relation of wavelengths to domain size, because then the advection component may also alter the pattern itself. And maybe one can make experiments in this way in comparing differently quickly growing plant embryos, if that is having an effect.
This is a very, very interesting question, and I can tell you something that I don't remember the result, but considering that the plant leaf, I think there is a Yu Yao paper about that, where they estimate 70–80% of the cell divide periclinally, so it creates somehow an advection term. And so at the beginning, when we start to play with the first model, we didn't take too much into account the difference between the rate of chemical reaction and rate of cell division. So if these rates are pretty similar, somehow you create an advection term. So also when the wavelength is shorter and you should make somehow variegate pattern, it tends to increase the robustness of the bipolar stripes, essentially. So the cell division could increase the robustness of the system, I would say. On the other hand, there is the fact that chemical reactions are much faster than the occurrence of division in plant. But on top of this, if not all the division occur in the proper angle, the plant also failed to make the proper flat lamina. You know, there is something that I couldn't answer to your question. So we know that the difference is not just simple in the orientation of cell division, but there is something that we should discover about what you asked that I don't have the answer now, but there is still puzzle underlying the directionality of cell division and the flatness of the leaf, but I don't have the answer right now.
Nice. And maybe if I can add another question, can you tell us more about the simulation environment that you've used and how that can be accessible by others?
We use Morpheus, and it's a platform that multi-level platform. Essentially, we start to make prototype in Mathematica, but once we need a little bit more power, we have this C++ developed environment. It's multi-level. We can make continuous model or cellular plot model in a very easy way and reproducible. So for the paper we share on Nature, this is a Nature Plant paper. You can go on the site, download the XML file, and with Morpheus you can reproduce the simulation really easily. There is also a page in Morpheus that describes how the model is built, and you can download the file and simulate on Morpheus. And that was for that. This I tell you that. We find compromise because in-house build model that completely reproduce the mechanical of the plant were super slow, while we had the advantage to use Morpheus and run thousands of simulation in a reasonable time, also because you can also run it on a server. And
Great, thank you.
And that's, I think, why I won the prize, because it was about the reproducibility of the model. So this means that anybody with one click can download and change the parameter and try to see if some botanicals that they have in the garden could be fitting with the phenotype produced by changing parameter of the model.
Nice, thank you.
And thank you, Lutz, for the question.
Other questions? I want to come back. Maybe you could tell us a little bit. I have two questions, really. One is coming back to the looking at all of the possible topologies of the regulatory network. So you said you started out with your 5 nodes, and then you have potentially 5 links between the node and all of the other nodes, and they could either be missing, activating, or inhibiting. So you have, I guess, 5 cubed possible networks. How did you go about? Could you remind me how you went about eliminating topologies that you were and coming up with this final selected topology? Did you look at that you had particular criteria for the patterning in the reaction diffusion model? How did you eliminate topological structures?
Okay, there is first of all, you have to eliminate the redundant or disconnected network. We start from 6. So maybe you can have a topology, but in reality, when you go to analyze, there are 2 topology completely disconnected. And but a part of the more let's say rule of topology, the first discern was topology that can make a Turing pattern, and also we were able to arrive only about up to 5 components, as you say. But we have the fact that the topology has to be done with 2 diffusible molecules, and these 2 diffusible molecules should produce an out-of-phase pattern. So one has to be in one domain and one in another. So we already go down to 130 topology. And after, there is the problem with the bigger topology. We have the problem that microRNA can have only one connection, that is the inhibition of the target. And maybe this is the reason why we didn't find any 4 and 5 component topology at the beginning. We have to go down to this module of 3, because the module of 3 is compensated with the transcription factor that we couple with the micro that could do the missing connection. Indeed, when we deconvolute, we discovered that one protein has to diffuse, and this make possible after that we have a minimal 4 component and a minimal 5 component topology. But this essentially the reason why we end up to a 3 component and only 1 is because there is a limited number of connections that we can have in the system.
But it's not that you're doing this essentially by thinking about what's possible, rather than, for example, by an exhaustive enumeration and then doing simulation and selecting for particular patterns.
No, we didn't do simulation. We used the mathematical theorem that has been developed by Turing, right, but essentially by later mathematician. There is work from Murray. There is also Weezy. There is James Sharp that published this mouse link paper that give us the link to this paper that was working on plant. There was a 3-component paper that explained really well how to make the constraint that we could have later run as a deductive system. Right. After that, we ran the first simulation.
Okay,
I think it was just maybe the fact that, as we were arguing early, plant cells do not move. There is not too much. We were pretty sure that it was done just by reaction diffusion. Otherwise, we could have go for a high throughput simulation and a way to discern from the simulation.
So the other question I had came back to the mechanics. You started out by saying you wanted a model that could explain flat leaves and also curled leaves and other morphologies. And the model so far, you have mechanics in the sense that when cells divide, they push on each other, but you don't actually have the full 3-dimensional You don't have the apical basal polarity of the cells. How would you begin to think about coupling a model of this kind to force generation if you wanted to do that?
Essentially, we are already thinking about that. There is a finite element model. We now, with the framework we have, we can easily predict the wavelength, for example. You can also predict without simulation, which is the wavelength of a linear system of Turing. And that could be a starting point. The fact that we fully focus on this is because without the patterning, the leaf is not flat. So we wanted to really discern this, and we didn't focus on the mechanic, because also simulating a finite element model, it costs a lot of iteration and it takes a lot of time to make just one simulation. So now that we are good in making a simulation of pattern, we are moving on with the mechanical part. And the essential part would be consider also other mechanical property, because there is the fact that there is a constraint that could feedback on that. So the first abstraction could be just introduce the patterning that change the mechanical property while it grows, but it's just one step, because we have also to consider the feedback of the mechanic on the gene expression, for example, or the biochemistry, that in plant are kind of really relevant. This is something that is emerging from several works.
Okay. Yes, well, that's something that could keep you busy for a long time to do the full mechanics of the plant. With the feedback in both directions. I wasn't trying to be critical. I was just interested in what your next steps were on that direction. Are there any other questions for our speaker for today? Again, if you need to unmute, please speak up in the chat and we can do that. Okay, well, I want to thank you then again for coming and speaking to us. I appreciate it.
Thank you. I appreciate the invitation.
This talk will be edited and put up online, so for people who want to view it again, or you want to share it with your colleagues who weren't able to attend today, it will be made available. Thank you, everyone, so much, and we will see you in 2 weeks.
Thank you very much.
Thank you. Bye.
Bye.